Alright, so today in class I had mentioned that we will be going over meiosis, and I want to make sure that what we did today in class was also put onto a video. It seemed like today really helped, so in case you guys wanted to go back and watch me go through meiosis one more time, I wanted to go ahead and make this video. So like we said today, we're going to start out with just a regular cell with a nucleus, and then we have chromosomes. Okay, and I'll explain a little bit more about these chromosomes here in a second. But what we have is we have 4 chromosomes. Okay, and remember we talked about 4 chromosomes in this cell. If it was a human, it would be, if it was a human, it would have 46 chromosomes or 23 pairs. Okay, and we can kind of look at the pairs, like this. Like that's a pair. This is a pair. Okay, so that's kind of what we're looking at here for pairs. And so what we get with this first part of mitosis is the first stage, which is, of course, we have interphase. Okay, interphase. And what happens in interphase is that the chromosomes duplicate. We get a copy. Okay, so each one of these chromosomes makes a copy of itself. And that's it for interphase. Okay, the cell grows and the cell makes these copies, and that is it for interphase. The next part, and I'll just draw a little arrow, is prophase. Okay, and during prophase, we have our nucleus starts to dissolve, and these chromosomes condense. Okay, some people might be wondering why I'm drawing them like K's. If you really wanted to, you could almost say that, let's just draw them like this. And so now they really look a little bit more, like X's, which is what everybody kind of thinks. But the K does just fine. It's really just, the original chromosome and its copy that are put together that kind of make it look like that. Okay, so there we go. There's prophase. Nucleus starts to dissolve. Chromosomes condense a little bit. And then what we have after that, after prophase, we have metaphase. Okay, and what we have in metaphase are we have our chromosomes again, and they have now lined up in the middle. Okay, but it's a little different here. It's not just, the chromosomes aren't lining up directly in the middle. The chromosomes are lining up in their pairs down the middle. Okay, so if you remember back to that first cell that we drew, we'd said that this was a pair and this was a pair. So the pairs are now lining up in the middle. And then we have these organs called centrosomes. And those organs are extending these spindle fibers out to reach out to the chromosomes. And there we have metaphase. Okay, and then right after metaphase, those spindle fibers really get to work, because in anaphase, our next stage, okay, I gotta remember to write these. Anaphase, then we have metaphase. Okay, so during anaphase, those spindle fibers are at work. Okay, and they are grabbing onto those chromosomes, and they have officially separated the chromosomes, the pairs of chromosomes, from each other. Okay, so the pairs of chromosomes have now been separated. After we have anaphase, we go into telophase. Okay, and in telophase we get our nucleus starting to reform. We have chromosome. We got both of our chromosomes, or should I say all 4 of our chromosomes. Okay, because if you remember, if we go back to our original one, we said that there were 4 original chromosomes, okay, in here. Come on, red. We said there were 4 original chromosomes in here. Now, if you look back, okay, again, we have 4. 1, 2, 3, 4. 4 chromosomes, okay, and after telophase, we have cytokinesis, where we get 2 individual cells, okay, 2 individual cells. I don't know what that is. 2 individual cells, okay, each with chromosomes. Each with 4 chromosomes. Okay? After cytokinesis. After cytokinesis. Now we do the exact same thing, except there's just one thing that's a little different. We're doing the same thing with 2 cells. So it's no longer just 1 cell, it's 2 cells. So we're back to prophase. Okay, but this is actually prophase 2. So we essentially have the same thing. Okay, but this time, not yellow. Okay, so it's the exact same thing. This time, it's just happening in 2 cells. Okay, then we have our metaphase. Okay, the exact same thing as the first metaphase. Chromosomes. They line up in the middle here. Remember we had said the last time that we've got these centrosomes, and what they do is they just grab onto these chromosomes. Okay, and this is metaphase 2. Alright, after that we just continue doing the same thing. Okay, this is going to be anaphase. Anaphase 2. Except this time, we're not just separating the pairs. Okay, we're not just separating the pairs. What we are separating here is we have separated the original chromosome from its copy. So if you remember, in the very first interphase, okay, in the very beginning, when we were talking interphase, and we said the very beginning of meiosis, we had said that the very first thing that happens is that chromosomes duplicate themselves or make a copy. And now what we're doing is we are separating the original chromosome from its copy. Okay, so if we go back and we look real quick, okay, and we said that these were the original chromosomes, and the part that was attached to it is the copy. If you go through the list, what we did was we separated the pairs, and now what we're doing is we're separating the original from its copy. Okay, and now we go to the very next part, which is going to be telophase 2, and in telophase 2, we get the same thing as telophase 1, except it's happening in both cells. Okay, nucleus starts to reform. Okay, except for this time, you can notice there's a little difference in the amount of chromosomes that each one of these cells has. Okay, so each chromosome, or each cell originally had 4 chromosomes, and now what we have is we have 4 cells, each with their own nucleus, and each having 2 chromosomes. Okay, so let's go through that one more time just to make sure. Okay, we start off with interphase. And this time we have 4 chromosomes. They double. That's why we get the extra kind of jagged parts on there. That's the double. Okay, prophase, the nucleus dissolves. In metaphase, the chromosome pairs line up in the middle. In anaphase, they start to pull apart. In telophase, they start to separate. And then what? Ooh, there's a mistake. Let's make sure that this chromosome is purple. Okay, they start to pull apart. The nucleus starts to reform. In cytokinesis, they completely separate. Okay, we have 2 new cells. And in prophase 2, when meiosis starts over again, it goes through the exact same thing with those 2 cells. So 1 cell became 2, and now we're going to watch this process happen again, and those 2 cells become 4. Okay, those 2 cells become 4, and each one of these has 2 chromosomes. So we started with 4, and we go to 2. Just like in humans, we start with 46, and we go to 23. And the reason that is because when you combine a female sex cell and a male sex cell, when you combine a sperm and an egg, okay, each with 23, when you combine those, you get 46. Okay, so hopefully this was helpful. Again, if you have any questions, please come and see me.